Hello students, welcome to EPG Pathsala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module that is fate of pyruvate under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We are going to understand what is fate of pyruvate under different conditions. We are also going to understand how pyruvate has three fates depending on the presence of oxygen. Under aerobic condition, entry into tricarboxylic acid cycle. Under anaerobic conditions, it undergo lactic fermentation, ethanol fermentation, and pyruvate decarboxylase system and alcohol dehydrogenase. Today, I am just briefly going to discuss the fate of pyruvate. So, the big question is what happened to pyruvate after it is made from glycolysis? Answer to this question is dependent on whether oxygen is present or not. Based on this fact, that is presence of molecular oxygen and the cellular metabolic need, pyruvate take up different routes for its metabolism. Pyruvate, end product of glycolysis, facilitates the production of lactate, alanine, acetyl coenzyme A, oxaloacetate, ethanol, and malic acid. This concept map gives the overview of fate of pyruvate. First, glucose is converted into 2 molecules of pyruvate by 10 steps of glycolysis reaction. In animal, plant and many microbial cell under aerobic condition, pyruvate is converted into acetyl coenzyme A, and further acetyl coenzyme A goes into the TCA cycle and completely oxidized into carbon dioxide and water. Under anaerobic condition, in yeast cell, pyruvate is converted into ethanol, and in vigorously contracting muscles, in red blood corpuscles, and in some microorganisms, pyruvate is converted into lactate. Pyruvate is a keystone molecule and it is critical for many aspects of eukaryotic and human metabolism. It is the end product of glycolysis. It is derived from additional sources in the cellular cytoplasm and ultimately transport into mitochondria as a master fuel. The fate of pyruvate depends on two factors, whether oxygen is present or not. On the basis of that, pyruvate undergoes different reactions. There are 3 reactions of pyruvate as a prime importance, such as conversion into ethanol, lactic acid, and acetyl coenzyme A. In plant, animal, and many microbial cell, glucose and other sugars are converted into pyruvate and ultimately oxidized into carbon dioxide and water via the tricarboxylic acid cycle and the respiratory chain. Before entering in the TCA cycle, pyruvate is converted into acetyl coenzyme A in the matrix of mitochondria by the enzyme pyruvate dehydrogenase complex, popularly known as PDH. It shows the mechanism of pyruvate dehydrogenase complex. The figure gives an idea about reaction mechanism of a PDH complex. The PDH complex is a key position in the oxidation of glucose by linking the glycolytic pathways to the oxidative pathways of the tricarboxylic acid cycle. In prokaryotes and eukaryotes organism, pyruvate dehydrogenase complex contain multiple copies of three catalytic enzymes: pyruvate dehydrogenase, enzyme 1, enzyme 2 dihydrolipoamide acetyl transferase, and enzyme 3 dihydrolipoamide dehydrogenase. In the figure, E1 is in the red color, E2 is in the green color, and E3 is in the blue color. PDH is also play an important role in degenerative neurological disease, obesity, type 2 diabetes, and other diseases. Recently, PDH has emerged as an enzyme of interest in cancer biology because of a switch from oxidative metabolism to aerobic glycolysis in some cancers. I could tell that some of you were not impressed by the mere 2 ATP produced per glucose by glycolysis. The 2 ATPs produced are only a small fraction of potential energy available from glucose. Under aerobic conditions, a much more dynamic pyruvate metabolism occurs. Under anaerobic condition, animals convert glucose into 2 molecules of lactate. Much of the potential energy of the glucose molecule remains untapped. In eukaryotes, what happens? All of the glycolytic enzymes are found in the cytosol. Charged molecule such as pyruvate must be transported in and out of the mitochondria. A small charged molecule with molecular weight of less than 10,000 can freely diffuse through the outer mitochondrial membrane through aqueous channel called porins. A transport protein called pyruvate translocase specifically transports the pyruvate through inner mitochondrial membrane into the matrix of mitochondria. Once it is mitochondria, pyruvate dehydrogenase convert pyruvate into acetyl coenzyme A and carbon dioxide. This irreversible reaction is the link between glycolysis and the citric acid cycle. This reaction may look straightforward, but it is very complex. Pyruvate dehydrogenase is a homogeneous enzyme that can be visualized by electron micrograph. Pyruvate dehydrogenase is a multi-enzyme complex held together by non-covalent interactions. There are 3 different enzymes and 5 coenzymes. The molecular weight of this pyruvate dehydrogenase isolated from E. coli is 4.6 million Dalton, slightly larger than a ribosome. Under anaerobic conditions, pyruvate is converted into lactate by lactate dehydrogenase enzyme. It takes place in muscles during intense activity. Mitochondrial pyruvate carboxylase catalyze the carboxylation of pyruvate to oxaloacetate, in which vitamin biotin is the coenzyme. Ethanol is formed from pyruvate in yeast and several other microorganisms. In first step, pyruvate is converted to acetaldehyde by pyruvate decarboxylase. In the second step, acetaldehyde is reduced to ethanol and reaction is catalyzed by alcohol dehydrogenase. Pyruvate is converted to malate by malate dehydrogenase, and it is an important source for the synthesis of NADPH. Pyruvate can be directly converted to oxaloacetate, or it is first carboxylated to malate and then decarboxylated to oxaloacetate. These two reactions are called carbon dioxide filling up reactions or anaplerotic reactions. They provide oxaloacetate when there is sudden influx of acetyl coenzyme A in the TCA cycle. In the absence of oxygen, that is under anaerobic condition, pyruvate undergoes fermentation, either lactic acid fermentation or alcohol fermentation. In this fermentation reaction, no ATP molecule is generated. In the lactic acid fermentation, pyruvate is converted into lactate by lactate dehydrogenase, and the alcohol fermentation is a two-step process. First, pyruvate is converted into acetaldehyde, and then acetaldehyde is converted into ethanol. Lactate fermentation occurs in human cell under certain conditions. During exercising, skeleton muscles are undergo vigorous muscle contraction and unable to obtain an adequate supply of oxygen, and therefore there are state of anoxia where muscle cells have to undergo anaerobic respiration. In these circumstances, skeleton muscle undergoes lactate fermentation to convert pyruvate into lactate. However, lactic acid is toxic to cells since it causes a change in pH, leads to acidosis and causing cramps and pain. The pyruvate made in red cells is converted to lactate by the enzyme lactate dehydrogenase. This is a slightly reversible reaction. During this process, NADH is oxidized to NAD+, which facilitate the reduction of pyruvate to lactate. If red blood cells have a lot of oxygen, then why pyruvate isn't converted to acetyl CoA and enters the TCS cycle? Because of lack of mitochondria. Lactate dehydrogenase is an important enzyme in human. If you see the mechanism of enzyme lactate dehydrogenase, it is a key enzyme in anaerobic respiration. Lactate dehydrogenase is responsible for the anaerobic conversion of NADH to NAD+. Lactate dehydrogenase is a quaternary protein formed of combination of two subunits, that is M and H, an abbreviation of muscles and heart, into a structure of four of the subunit. There are various combinations found in the human body. These combinations are 4H in heart, 3H 1M in reticuloendothelial, 2H 2M in lungs, 1H 3M in kidney, 4M in muscle and liver. Lactic dehydrogenase enzyme comprise 40% alpha helices and 23% beta acids. The SCOP data classify this form of lactic dehydrogenase as mixed beta alpha beta with mainly parallel beta sheets. Once the NADH is bound to the enzyme lactic dehydrogenase, then possible for lactate to bind substrate oxamate is shown. The CH3 group is replaced by NH2 group to form oxamate. Lactate bind to the enzyme between the nicotinamide ring and several lactate dehydrogenase residue. Then transfer of a hydride ion happens quickly in either direction, giving a mixture of the two tertiary complex enzymes NAD plus lactate and enzyme NADH pyruvate. Finally, pyruvate dissociate from the enzymes and followed by NAD plus. Ethanol fermentation. The other anaerobic fate of pyruvate is alcohol fermentation, which occur in yeast cell. Here NADH is regenerated by the fermentation of pyruvate to ethanol and carbon dioxide. Two molecules of pyruvate are converted into two molecules of ethanol and two molecules of carbon dioxide. It is a two-step reaction, and we use two types of enzymes that are pyruvate decarboxylase and alcohol dehydrogenase. And TPP is a cofactor for both these enzymes. First, pyruvate is converted to acetaldehyde by the enzyme pyruvate decarboxylase. This is an irreversible reaction. Pyruvate decarboxylase is an enzyme which decarboxylates pyruvate, which means the carboxyl group of pyruvate lose a carbon dioxide molecule and results in acetaldehyde, which serve as the substrate for the next enzyme in the pathway. Pyruvate decarboxylase require magnesium ion as a cofactor and also needs TPP as a coenzyme. TPP or thiamine pyrophosphate is derived from thiamine, that is vitamin B1, and contains a thiazolum ring which acts as an electron sink at the second carbon of pyruvate. Alcohol dehydrogenase is the other enzyme required in this process. This takes acetaldehyde and NADH and form NAD plus, ethanol and carbon dioxide. It should be briefly noted that lactate and ethanol are not the only product of microbial fermentation. Mainly, other commercially useful products are created and exploited. Mechanism of enzyme pyruvate decarboxylase, that is PDC. Pyruvate decarboxylase is a homotetrameric enzyme. Enzyme classification number is 4.1.1.1 that catalyzes the decarboxylation of pyruvic acid to acetaldehyde and carbon dioxide in cytoplasm of prokaryotes and in the mitochondria of eukaryotes. It is also called 2-oxoacid carboxylase and alpha ketoacid carboxylase. In anaerobic conditions, this enzyme is part of the fermentation process that occur in yeast, especially of the Saccharomyces genus, to produce ethanol by fermentation. Pyruvate decarboxylase start this process by converting pyruvate into acetaldehyde, and carbon dioxide. Pyruvate decarboxylase depends on cofactor thiamine pyrophosphate, that is TPP, and magnesium. Each identical subunit of pyruvate decarboxylase consists of approximately alternating alpha helices and beta acids, and two domains exist with each 60 kilodalton subunit. The active site of pyruvate decarboxylase in Zymomonas mobilis consists of GLU 50, GLU 473, ASP 27, and HIS 114. Hydrogen bonding occurs between the substrate and ASP27, HIS 114, and THR72. Yeast has similar active site residue. In the catalytic step of the reaction mechanism, the amino acid at 473 position donate a portion to the pyruvate. The negative charge of amino acid residue followed the protonation of the substrate lead to destabilization of pyruvate carboxylate group. Next, the carboxyl group leaves following decarboxylation. In the final step of the mechanism, release the acetaldehyde. A proton is transferred to the amino acid on position 473 residue from a cofactor. After the protonation, in a concerted step, water molecule donates a proton to substrate while receiving a proton from 473 number amino acid. As the proton is taken from substrate, the electron moves to form a carbonyl, which leads to the release of acetaldehyde. This pyruvate decarboxylase enzyme is regulated by substrate activation. This means if substrate is not present in the pathway, the protein will be off. The residue that is bound to start a cascade of events resulting in the activation of enzyme. This process allows the enzyme to be on when function is necessary and off when it would not be catalyzing reaction even if it were on. Mechanism of alcohol dehydrogenase enzyme. Alcohol dehydrogenase enzyme classification is 1.1.1.1 are a group of dehydrogenase enzymes that occur in many organisms and facilitate the interconversion between alcohol and aldehydes or ketones with the reduction of nicotinamide adenine dinucleotide to NADH. In human and many other animals, they serve to break down alcohols that otherwise are toxic, and they also participate in generation of useful aldehyde, ketone or alcohol groups during biosynthesis of various metabolites. In yeast, plant and many bacteria, some alcohol dehydrogenases catalyze the opposite reaction as part of fermentation to ensure a constant supply of NAD. The active site of human alcohol dehydrogenase consists of a zinc atom. In the community study, if you see in the commonly horse liver isoforms, the amino acid number 48 is a serine and leucine on 319 position is phenylalanine. The zinc coordinate the substrate. The zinc is coordinated by cysteine 46 number, cysteine 174 number, and histidine 67 number. Leucine on 319, alanine on 317, and histidine on 51, and isoleucine on 269, and valine on 292 stabilizes NADH by forming hydrogen bonds. Histidine on 51 number and isoleucine on 269 number form hydrogen bond with acetaldehyde on nicotinamide ribose. Phenylalanine on 319, alanine on 317 and valine on 292 form hydrogen bond with amide on NADH. In nutshell, pyruvate is keystone molecule and it is critical for many aspects of eukaryotic and human metabolism. It is derived from additional sources in the cellular cytoplasm and ultimately transport into mitochondria as a master fuel. Fate of pyruvate depends on whether oxygen is present or not. On the basis of that, pyruvate is converted into different byproduct. So students, let us summarize what we have learned in this module. Pyruvate, which is the end product of glycolysis, must be further metabolized to maintain proper redox balance. We understand the different reactions in which pyruvate undergoes under different cellular conditions and in various cell type, and to know biological and clinical significance of each of the reactions. Pyruvate is a keystone molecules and it is critical for many aspects of eukaryotic and human metabolism. It is derived from additional source in the cellular cytoplasm and ultimately transport into mitochondria as a master fuel. Fate of pyruvate depends on whether oxygen is present or not. On the basis of that, pyruvate is converted into different byproduct. So this is all about the fate of pyruvate. Thank you.